Hello, curious minds, and welcome back to another episode of the HEAS podcast. I'm your host, Victoria, and I'm thrilled to be back with more science knowledge and two new amazing guests. Today, we're diving into the fascinating world of ancient genomics and primate social behavior with two leading scientists from the Max Planck Institute in Germany, Kelso and Tung. Our first guest, Kelso, is an eminent figure in the field of ancient DNA. Janet is the group leader of the Ancient Computational Genomics Department at the Max Planck Institute, and she was involved in so many cool projects. Among them, the sequencing of the first Neandertal genome, the analysis of a completely new hominin group called the Denisovans, and so much more. In our conversation, we explore her journey, the challenges of working with ancient DNA, and the insights these studies provide us about our closest hominin relatives. As you will notice in the beginning of our conversation, we're just diving right into it. And that's because I had already started recording before starting the official part of the podcast. And it was already such a good conversation that I single-handedly decided to keep this part in as well. Our second fabulous guest is Tung. Jenny is the director of the Department of Primate Behavior and Evolution at the MPI, Max Planck Institute in Germany. And she's also a professor of evolutionary anthropology and biology at Duke University in the US. Her research focuses on how social environments impact the health and mortality in primates. We talk about how the social environment of primates is predicting their health and mortality, probably even more so than their genetic makeup. We also discuss the vital roles of social relationships, especially the importance of a mother figure in primate survival. We also discuss the surprising benefits that grandmothers provide to their grandchildren in baboon societies. Let me say this. It was a pleasure to sit down with both of you, Janet and Jenny. And I really have to say, I totally see why both of you guys are so successful in what you're doing and also good friends, which I love to hear, because not only are you extremely smart, you also have great personalities. So I want to say thank you once again for taking the time to talk to me and for motivating me, and I'm sure all of the listeners, scientists, listening to this episode. And I thought I'd drop that once more here. I would really love to come see both of your labs at the MPI. Just saying. And with that, I'll leave you, my listeners, to this educational and entertaining episode. And as always, stay curious, stay engaged, and welcome to the HEAS podcast. I got a bit nervous. When I saw you were coming and you were on my list of people that I will be interviewing, I was like, oh my God, I really have to get my shit together for that one, you know?
Oh, no.
And then from the talk, I was like, oh my God, I was frantically typing. I was like, there's so much I do not know.
So you have to do a whole lot of research as well. So you really spend time reading.
Most of the times I really try to on the weekend.
It's a lot of really different things.
It is. So the archaeological
What's your background?
Yeah, I'm an ancient DNA.
That's what I thought. But people in ancient DNA come from different fields. So you're an ancient DNA person, right?
Yes. But I do interview people from landscape archaeology and stuff. And I'm always like,
I saw some of the previous speakers and I was a bit kind of.
Yeah. Okay. But still, I mean, I still have so much to learn. Even in ancient DNA, I'm always like, that I didn't know, that I forgot.
Me too.
Okay.
Plenty to learn all the time.
Always. Yes. I feel like when you start working in a certain field, there's like this this very steep learning curve, and then you're like, oh, I'm already a bit, sophisticated, and then it starts like to crumble, and you're like, I don't know shit.
I don't know. I think there's a point where you realize what you don't know, and that's when you really realize. That's when you know that you've kind of come to the field, right? When you realize what it is that you don't yet know.
Yeah. That's a reality check.
It is. And I read papers often that I think, oh, wow, that's a very clever approach. I wouldn't have thought of that, or I didn't know that was possible. It's still really, I still get this feeling often, right? There are many things.
Yeah.
Ancient DNA is such a broad field.
Yes.
That, we're taking approaches from environmental biology, from
even more
computational biology, genomics, and I think as a result, it's hard to be a specialist in everything.
I mean, if you are, then something must be wrong.
That could be.
Okay, I think we should officially start this conversation because maybe I can cut that in because it was already so good. I will be reading a bit more than usual because I don't want to mess up my questions. They're a lot longer than they usually are because I want to get the juicy scoop from you. So, are you ready? Welcome to the HEAS podcast, Janet.
Thank you very much. I'm very happy to be here.
I'm so honored to be able to sit down and chat with you today. You have no idea. First of all, let me say you're a huge deal in ancient DNA and a great role model for other female scientists like me, because what you do not know is that many of your papers, especially the earlier ones from the 2010s, have definitely inspired me and played a big role in me wanting to study anthropology and human evolution, which years later I did. So could you please introduce yourself and your amazing job?
Sure. First of all, it makes me really happy that work that I've done in the past inspires other people. I guess you don't normally do work thinking that you'll inspire other people. You do it because you're interested in it, but it's a nice side product. So my name's Janet Kelso, and I'm a computational biologist who works in ancient genomics, and I work at the Max Planck Institute for Evolutionary Anthropology, which is in Germany. And I've worked there for 20 years now, which feels like a really long time, and it went by in a blur. My job, I am a group leader at the Max Planck Institute for Evolutionary Anthropology, a group leader in ancient computational genomics. And that means my group applies computational methods to the analysis of DNA from ancient samples, mostly ancient human samples. And so we work on Neanderthals, and we work on Denisovans, and we work on very early modern humans. We like to focus on humans that are older than 30,000 years. That would be, I guess, our happy space. That's, I think, a good explanation.
As you said, you joined the Max Planck Institute for Evolutionary Anthropology as a group leader in 2004 already, at not even 30 years old. Correct me if I'm wrong.
That's correct.
That is
27. No, 29.
29.
Yeah.
That is so impressive, Janet. And as we did chat a bit before, you mentioned you're the same age as most of the students. I mean, what's that like?
So I feel very fortunate to have to be offered. Felt very fortunate to be offered the position. I was certainly intimidated when I first got there. But this was an institute, at least in 2004 when I joined. I didn't join to work on ancient DNA. That wasn't actually part of my job description. So at that time, my background was in gene expression analysis, and I'd been doing computational work on understanding gene expression. And at that point in time, using primate DNA, right? So we were using comparison to chimpanzees, between chimpanzees and humans, to understand human-specific changes in gene expression. And I think in 2004, this was still prior to the time when it was possible to really imagine sequencing whole genomes of ancient humans. So the nearest living relatives we had then were the chimpanzees. And so I was hired to be a group leader in bioinformatics and to work on gene expression comparison with a team that was then at the Max Planck Institute. The ancient DNA came later.
As you said, it was not even imaginable that we could sequence ancient hominids, because can you tell our listeners when the first human modern genome was sequenced, to put that into perspective?
So I think the publication, I'm going to get this wrong. I think the publication of the first modern human genome sequence was in 2005.
So around that
Right?
time.
I think it was around the same time.
Around the same time. That's crazy.
And that was a massive effort, right? That was an effort of tens of labs worldwide, one human genome sequence, tens of labs worldwide, millions of dollars invested in doing this. But that whole project really inspired massive enhancements and kind of innovations in sequencing technologies. And the companies that are now sort of the world leaders in providing sequencing instruments for sequencing DNA, they come out of this time period where people were thinking, how can we sequence more than one human genome and for ideally less than millions of dollars, right? So this idea that you could sequence populations, which simply wouldn't have been feasible with the sequencing technologies available prior to 2007 or so.
So probably over the last 15 years, your lab and the whole institute contributed some groundbreaking discoveries. And for example, there was the publication of the first Neanderthal draft genome in 2010, the discovery of a completely new ancient hominin species, the Denisovans, from the Denisova cave in the same year, so 2010. And then another great publication of a hybrid, a Neandertal Denisovan hybrid. Just to name a few. So what stood out to you personally? What was your most outstanding moment or project during your career at the Max Planck Institute?
It's a really tough question. I think the first Neanderthal genome will always be a very special memory. I mean, it was a project that we worked on over quite some time before it was published. So I remember sitting around the table in Leipzig discussing, do we think it would be possible to sequence a Neanderthal genome given these new sequencing technologies that are coming on the market? At that time, there was a sequencing technology called 454. And we were talking with the developers of the technology about whether it might be possible to think about sequencing a Neanderthal and actually work together with them in a collaboration to start the sequencing. But the discussions at that time, I have to say, we were optimistic, but also the calculations that we did of what the costs would look like and how much bone material we would need and how much DNA we would have to actually extract, they were quite sobering. So it was a very inspirational time. It was a great team of people. And I think what I remember from the time mostly is the sitting around. If you've been in the Max Planck Institute in Leipzig, you'll know in the genetics department, there's this big table. It's a big seminar area, and that's where all the meetings happen. And the Neanderthal meetings were on a Friday afternoon, and they started at 1:00. They frequently ran through dinner. So we would go and have dinner together down in the cafeteria, continuing to talk science, maybe at 6 or so, and then we would go back and continue our meetings. So we had meetings that ran all the way into late on Friday evenings, as only scientists can, right? We don't have anything else to do. This was the coolest thing that we could be doing at the time. We had an amazing set of collaborators, so they were online on phone conferences with us. But just this feeling of, we don't know if this is possible, but we're going to try. I think that project is very special. And then, of course, if I had to choose a second favorite project, it's the one I'm working on now, because, of course, the thing you're currently doing is somehow always the most exciting. And so that's working on new Denisovans and trying to understand a bit more about the variation that existed among these previously unknown group of humans.
Working on Denisovans is not the same as working on Neanderthals, because Neanderthals were first identified as a new species through plenty of fossil remains, quote unquote plenty. Of course, there's not a lot as modern humans, but more than Denisovans, definitely. So this was not the case for the Denisovans, because that's a hominin species that was found with just a few fragments found at Denisova cave in the Altai Mountains in Russia. So what we have is a couple of tiny bone fragments. Among them, there is a toe bone, a tooth, and then a mandible from China. So that's from a different site. So we don't have a lot of material. And the Denisovan ancestry was, however, only determined through molecular analyses and not morphological ones. Do you think we will be able to identify other hominin species, maybe even older ones, from a molecular level in the future?
I think that's I think it's possible, yes, of course. I mean, if you can extract DNA from a bone or a tooth, or even more recently from sediments, it's possible to identify new species. I mean, we did not expect to identify the Denisovans. It was sort of a very fortuitous finding.
Could you tell us more about that finger bone that was discovered at Denisova Cave?
Yeah, so our collaborators at Denisova Cave, the Russian archaeologists, so Anatoly Dorovenko and Mikhail Shunkov, found this finger bone. And they asked us if we would, well, they asked Svante Pääbo if he would like to look at it molecularly. And I think what they thought was that it was probably a Neanderthal or maybe a modern human. It's found quite a recent layer in the stratigraphy in the cave, so in the layers of the cave. What we didn't know at the time was that that bone had been bigger originally. So we received a very small piece, and we initially sequenced a low coverage genome from that individual, so just a little bit of DNA from it. And because it was so interesting, right, because it was clearly a new hominin group, we wanted to sequence more. And so Svante went back to our collaborators and said to them, is there any more material? And they said, oh, there was this other part of the bone that they sent to another lab. And we said, wait, another part of the bone? And as far as we can tell, that bone went to a lab in the US. We don't exactly know how, but it landed up in a lab in France, was then scanned there. So we actually have an image of it. But we don't have an image of what the whole original bone looked like before it was cut. So it was cut. We were given one piece. They were given another piece. The lab in France has now published the scans, the reconstruction of the full bone, so a computational reconstruction of the whole finger bone, and also the genetic data from the bone, showing that it really is the same individual. It's the other part of the bone. So the bone itself has a really interesting story. But we didn't get any more, right? So we didn't get the rest of the bone. And so we turned to a different approach. And that was one of my colleagues, who's a biochemist, worked on developing a new method for improving how much DNA you can get from an ancient sample. And he developed this so-called single-stranded method, which basically means you take the DNA fragments that you have and DNA is double-stranded, you split the two strands apart and you capture both of them. So you have a chance to see both strands. And that really increases the amount of DNA you can get from old samples. And by doing that, we were able to actually sequence the 30-fold. So 30-fold genome means that we see every position in the genome on average 30 times, right? Mm-hmm. So we really have high confidence. If we see a base, we're pretty sure that that's that base. And so that was what made the high coverage genome of this Denisovan individual possible, actually, because we thought we couldn't get any more material.
But still, it turned out fine. You got a high coverage genome from that little bone. And do you remember this moment of realizing it doesn't fall within the modern human variation, and also it's not a Neanderthal?
So the work on this was done, the initial work on this was done with a mitochondrial genome, and it was done by a graduate student at the time, Xiaomei Fu, together with a postdoc, Johannes Krause. And so they were the ones that made the original discovery, right? And they were the ones who saw this, and I guess, like all scientists, they saw and said, well, that's weird. Like, maybe I made some kind of mistake, which I think is what we often think, right? Something looks weird, it's not what you expect, and you think, could this be an error? Did I do something wrong? That's very much a recurrent theme, I think, in some of our analyses. And then you do all the checks to make sure, right, you did the analysis correctly. And so I wasn't present when this discovery was made, but I've heard about it since. And there was apparently a phone call made by Johannes to Svante saying, we have this very interesting observation, the mitochondrial genome of these Denisovans. It's outside of, it diverges, it's much more deeply related to Neanderthals and modern humans than anything we've seen before. It looks like something really different, a different kind of human.
That must have been a really amazing moment. And so having seen these results and the advancements in these methods, do you think we could at some point get Homo erectus DNA? I know they're a lot older. Could you give us some dates for Denisovans? So that we know.
Yeah. So the Denisova, what we call Denisova 3, the Denisovan genome that that we were just talking about, that comes from an individual that's dated to between 50 and 60,000 years old. It's, what we would call one of the late Denisovans, one of the youngest Denisovans. We have Denisovan DNA from much older Denisovans too. So we know that Denisovans at Denisova Cave were present there already at least 200,000 years ago. And we know that from the genomes of additional fossil material and also from DNA that's been found in the sediment, in the sand at Denisova Cave. And sort of layers of the cave show evidence for Denisovan presence in the cave over an extended period of time. So starting around 200,000 years ago, or a bit more than that, all the way until around 50,000 years ago.
And other species, like I mentioned, Homo erectus, it's just an example, but they're a lot older. So the chances of retrieving ancient DNA is probably decreasing with age and with other factors, as you've explained prior in your talk, temperature and all that stuff. So I guess there is, I would say, I hope there is a potential, but what's your gut feeling?
I hope so too. I don't know about my gut feeling, but I really hope that it would be possible with enough sampling, with enough, with a bit of luck, which I think often plays a role here. A bit of luck, a lot of sampling, a lot of hard work. It could be possible to get DNA from, I'm going to say, other forms of humans, whether Homo erectus or we know about other forms of humans that lived at the same time as Neanderthals and Denisovans, things like Homo luzonensis, and in Luzon in the Philippines, the hobbits, of course, Homo floresiensis in Indonesia. I hope that at some point we would have DNA from at least some of these.
You're right. That would be even more realistic because they're not that old. And
yeah, but they're in a very bad area for DNA preservation. They're in all of them in island Southeast Asia, which of course is a really interesting place for human evolution, but it's a very bad place for DNA preservation.
As I learned, because I screened so many caves across the globe, and there's just nothing to find there for ancient DNA in those areas. So let's move back a little. As you said, you and your team are computationally studying and comparing the genomes of modern humans and also their close ancestors. To set into perspective the genomic relatedness between our closest hominin ancestors and us, let's begin with a little background information. So let's go back into the phylogenetic tree. Chimpanzees and humans had their last common ancestor about 6 to 8 million years ago. But still, they share 99% of their DNA. That's a striking similarity if you think about the time. Now, how closely related are we to other extinct hominin species that fall even closer to us on the phylogenetic tree, like Neanderthals and Denisovans? Or should I rather ask, what are the molecular differences?
So you just said that we split from a common ancestor with the chimpanzees about 6 million years ago. By comparison, our last common ancestor, the split from our last common ancestor with Neanderthals and Denisovans, is on the order of 600,000-700,000 years ago. So we're much more closely related to them than we are to chimpanzees, right? They're our nearest relatives. They just happen to be extinct. That means that, so we're sort of 99.8% similar to Neanderthals and Denisovans, right? So we're equally closely related to the two of them.
It was just very striking to me that if you look at the phylogenetic tree and you see chimpanzees, and there is so many obvious differences behaviorally in everything we are that separates us from what they are. And we can still watch their behavior and study this. But compared to other extinct species that we cannot study in this way anymore, it's striking that we still share 99% of our DNA with chimpanzees, and then how closely related must we be to them? And what is interesting to me in that aspect is that we always see ourselves as like more evolved, so much, in a way better, as lack of better words, but we must be so similar in a way to them.
I mean, I think that it is sort of a very human thing to think of ourselves as somehow superior. We've
Superior.
evolved for exactly the same amount of time as chimpanzees are, right? So chimpanzees are evolved for just as long as we are. They're evolved to suit their habitats. We're evolved to suit ours. I think what is an interesting question is there a genetic basis for some of the traits that differ between us? And we know that for some simple things there are, right? So we can describe the genetic basis of things like having a hairy body, like a chimpanzee, right? That's sort of obvious, but then it gets a bit more complicated. Even now, people debate what really is the difference to chimpanzees, right? There's speech and language, but chimpanzees also communicate. It's not that they don't have a method of communicating. There's upright walking, but they do that too.
They do that too.
There's cooperative behavior. Chimps hunt together too, right? So the question is, what is it that distinguishes us? And it's a complicated question, and I think not one that is simply answered with, here are 3 genetic variants that explain the differences between. I think many people would say, oh, we're interested in what are the differences between Neanderthals and modern humans, or between chimpanzees and modern humans. And to make a list of the genetic differences, that's quite easy, right? We can compare the genomes of the Neanderthals and the Denisovans and all modern humans, and we can pick out places where modern humans carry specific variants. And we can say those are candidates for making us special. But we then have a rather long list, and it's very hard to go from that long list to what it means. What does it mean phenotypically? What does it mean for our behavior? Even more complicated. And that's the subject of research. And it's the subject of research even among people living today, right? What is the genetic basis of differences in behavior between people living today? Is not a question that we can answer mostly yet. And so I think that it's a big challenge, and it'll be a big challenge for a long time still to understand, can we predict maybe how Neanderthals or Denisovans were in terms of their cognitive abilities, in terms of their behaviors, from their genetic sequences? Right now, I couldn't do that if I had your genome sequence. I couldn't predict your cognitive abilities or your behavior. So I think it's a very tall ask to be able to do that for archaic humans.
Those are the big questions that are still remaining.
Yeah, absolutely.
But what we do know from their behavior is that Neanderthals and anatomically modern humans did get together quite frequently and did produce offspring. So Neanderthals did go extinct approximately 40,000 years ago, as we know. And we know that present-day non-African people still carry approximately 2% of Neanderthal DNA in their genomes. And we do also know that Oceanians carry approximately 4% of Denisovan DNA, with higher percentages found in certain areas, I think, like the Philippines, Australia, and Papua New Guinea.
Yeah, exactly.
Given these facts, can we deduce that mating between modern humans and other hominin species was a frequent and common thing?
I think that's a great question. So I think when modern humans and archaic humans met, they interbred. We have some evidence of that, right? We have the evidence that everyone outside Africa today whose ancestors met the Neanderthals carries Neanderthal DNA. So all of us have 2% Neanderthal DNA. We also see that early modern humans in Europe, the earliest modern humans in Europe, 4 out of the 5, I think, earliest modern humans in Europe carry Neanderthal ancestry, additional Neanderthal ancestry, meaning that they met Neanderthal populations again after this first mixing, and they again mixed with them. So yes, I would say mixing was common when they met, but we also think that Neanderthal populations were potentially quite small and probably quite widely dispersed across Eurasia. So it may not have been that they met that often, which is why we don't see, multiple recurrent waves of integration or mixture with Neanderthals in the genomes of people living today.
It's hard to think of a world where not that many humans were populating the Earth. But in that case, so when we met, we did mate.
I think so. I think that that's kind of the emerging picture. And I mean, there's been a lot of discussion about why Neanderthals went extinct. And maybe in some way one could say that they didn't really completely go extinct, right? We sort of moved in, absorbed them into the modern human population. Some of them died out, but we absorbed some of them into the modern human population. We all carry Neanderthal genes with us. So they're extinct, but we're carrying sort of a good proportion of the Neanderthal genome around in people living today. If you sort of take pieces of Neanderthal DNA out of all of our genomes, you can reconstruct about 40% of the Neanderthal genome.
Oh, that's
just from people living today.
Yeah. 40%. That's amazing. I also like that point of view that we kind of integrated a bit of them and we keep bits and parts of them.
There was sort of like this really, I sort of think of it as this really big imbalance in population sizes, right? The Neanderthals seem to have lived in quite small groups. They were probably quite a small population. Modern humans at that point in time begin a huge expansion as they enter Eurasia. So they're larger groups and just integrating in, I think integrating in some combination of integrating in Neanderthals and some Neanderthal populations just going extinct is probably closer to the reality than anything else.
Thanks for explaining that. I think that gives a really good overview on what could have happened during that time, which is still a period of a lot of speculations.
Absolutely.
So there is a certain tradition at the Max Planck Institute involving a pond on campus.
The slimy pond.
Oh, is it slimy? To give some context, PhD students, I think, are thrown in there after receiving their PhD, after finalizing. And so was Svante Pääbo, after receiving his Nobel Prize in 2022. And Svante, for some background info, is basically one of the founders, the founding fathers of paleogenomics, and he's also the director of the institute in Leipzig. After all the success you're having and the impactful papers you're publishing, are you afraid you could land in there as well at some point?
I hope not. I purposefully didn't get my PhD there. Yes, this is really a tradition in genetics. We throw PhD students in the pond. We also throw Nobel Prize winners in the pond. Now that's a new tradition. So I think I'm safe for now.
Oh, I wouldn't say that, Jenna.
We have another tradition, though, and that's that everyone who graduates in our department gets to write their name on a big wall, one of the walls in the department. So we have this beautiful wall with the sort of memories of all the past PhD students and their signatures on the wall. And I'm very much hoping that I will never be thrown in the pond. It's pretty slimy in the summer and very frozen in the winter.
Oh my. So the summer then still is the better option, I guess, if you don't want to break your hip.
Yeah.
Maybe, I don't know.
We had a PhD student graduate in winter, and there was a discussion about whether we should dig a hole in the ice, and that was forbidden. So we just poured water over her.
Great stories. Thanks for sharing. Let's move on to our listener questions. I do go on Instagram and collect some questions from our followers there, and I'm going to ask you some of them right now. What are the ethical considerations involved in ancient human DNA research?
It's a good question. There's been increasing awareness that it's important to consider ethics when dealing with ancient human remains and data. At the moment, that is primarily focused on ancient modern humans, people who are direct ancestors of people living today. So, and I think that's perfectly appropriate and important. And there's quite a extensive discussion in the field of ancient DNA about how ethical permission should be obtained to sample ancient human remains and how data should be handled and to what extent the communities that are descendants of the people being studied should be involved in the studies. And I think that's an important discussion. It's not something where everyone agrees, but it's certainly an important discussion. And there's certainly more awareness of it. This discussion doesn't touch our research as much because we're working on Neanderthals and Denisovans who are not direct ancestors or immediate ancestors of people living today, right? They're not related in the family trees of people living today. So there's no one who claims that they're sort of their property, if you want to call it that way. That said, I think there's another kind of ethics that's important, and that's the ethics of sampling. So these remains are precious and often quite rare, and we have a responsibility to sample them carefully, trying to do as little damage as possible, so to take as smaller samples as we can from as unobtrusive a way as we can, and then to generate good data from those samples, because that's the only way that they become useful to then a much wider group of people. So, and we take that quite seriously. So we follow a sampling protocol that starts by taking what we call microsamples, very small amounts, ideally from not visible places in bones, if possible. We then perform our very best efforts to get DNA from those samples, and that often involves many rounds of experiments with the bone or tooth powder that we get. And once we have data, the final part of that responsibility is making that data public so that there's a benefit to the largest possible group of people, right? And so that people don't have to go and sample again, potentially destroying the fossil remains even more. And so for me, that's for this very old time period, the ethics that matters a lot.
That is so right. While I was asking that question, I thought, in the beginning you mentioned that you're dealing with samples or specimens that are older than 30,000 years old. So that doesn't totally apply in the sense that probably like repatriation, is that the word?
Yes. Yeah.
Yeah, you cannot do that because who would you repatriate it to? There is nobody left. But in that sense, you're totally right. We have to be so considerate on how we sample and that we get the best out of it. And then in the end, share this with everyone who wants to make use of it.
Exactly.
And then can incorporate the data in further and follow-up studies.
I mean, for us, one of the important things is just to try and minimize the amount of damage to the material, right?
And that's where sediment DNA comes into play.
Exactly.
Yeah, which is also a really, really cool and quite new field.
Yeah.
Where you can obtain, as you mentioned, traces of DNA or with the advanced methods, even more DNA so that you can even analyze do population genetics with this obtained data from sediment without damaging an actual fossil remain. Yep. So you can answer that, but I'm sure you will not be unbiased.
Okay.
What's it like to work with Svante Pääbo?
It's 20 years of a really great work environment. I sort of feel very fortunate because the work environment that Svante creates in the genetics department is one where everyone has a voice and there's a lot of discussion, a lot of openness. The teams are very integrated. I think perhaps a little unusually in a normal environment, often research groups are quite isolated from one another. In our department, that's not the case. The research groups work extremely closely together. And I think it's not just nice and collegial. I think it actually leads to being able to make advances more quickly. Because if you have biochemists and molecular biologists talking to computational biologists every day about a project where you really work head to head constantly on the project, you can identify problems and solve them much faster than if you say, well, I work in my silo, you work in your silo, you generate the data, I'll analyze it. We can think about how we're going to analyze the data before we generate it and make sure we generate it appropriately. And I think that's been a huge competitive edge for the culture that Svante has built in the department in evolutionary genetics. The sort of extremely open, extremely friendly, but competitive in the sense that we want to get this out there. We want people to know what data we're generating, and we're interested in the answers to the questions we're asking.
I was never lucky to visit your institute because, many of the PhD students, the other PhD candidates that I know, went there for like a week or two or so. But I started during the pandemic, then we moved buildings, and now I'm here. But yeah, I'm happy I get to talk to you and also other people from that institute.
The opportunity exists. The institute's not that far away. It's a short train ride. You can visit anytime.
I would love to, honestly. I have 4 months
You're
left.
You're very welcome.
Thank you so much. Let's move on to the lightning round. So it's called the Hayes Quick Take. So I'm going to ask you a couple of questions and just answer with the first thing that comes to mind.
Let's see.
All right, Janet. If you got a plane ticket right now, where would it go to?
I'd go to South Africa. I'm from South Africa. It's an amazing place, not only because I come from there and because I love it very much, but it's got a hugely rich hominin archaeological history too. And so interesting new discoveries of old hominins like Homo naledi. It's a great place. I would like to go and visit some of these sites and, of course, see my family.
Of course. What's a talent of yours?
A talent of mine. I think I'm good at reading and writing. So kind of writing.
I mean, I'm sure
that's a
you
weird
are.
thing.
We've seen
It's a weird
that.
thing to
but
say. I don't know.
Like outside of science.
Outside of science.
You're good at
You
that,
want to know
right?
something like,
I want to get the
outside of
scoop.
science.
We know you're good at science.
Not good at science. It's a particular talent. I'm very good at baking. That's it.
Oh.
I love baking. I don't get to do it nearly enough. I enjoy it very much. It's my relaxation. Trying out new recipes, and then feeding people in my family or neighbors or
Lovely. Lovely. I
with
mean,
things like that.
We all here in this department love the people who always bring cake. Shout out to Florian. Thank you for the great cake. I'm waiting for the next one.
Yep.
Yeah, that's a great talent. I'm not very good at it. And yeah, I kind of stopped. I had a phase where I was really into it, but
I so enjoy it. I just don't get enough time to do it.
Yeah, I can imagine.
It sort of inspire. Your question inspires me to think about doing it more often. That's a very hard question. Wow. I didn't think it would be a hard question. Then I'm like, I have no idea what I'm good at. Self-doubt creeps in.
Yeah, there we are, back at it again. Do you have a favorite mantra or quote?
Just do it.
Yes.
For me, if people ask me, so what's the trick to like going interesting places, doing interesting things? It's just take every opportunity that comes your way. Just say yes. I often tell my students, should I go for this interview? Just say yes. You can go. Never turn down an opportunity.
I love that so much because it's also my dad's favorite mantra.
Oh, yeah.
He says, there's nothing more powerful than just do it. Nike really hit it on the head.
Yeah, that's absolutely true.
If you could time travel, which era would you visit?
Oh, the Paleolithic. I want to see Neanderthals. I want to see Denisovans. I'd love to be at Denisova Cave.
Oh,
I'd love to see how it was used. Who was there, if we were right about our ideas that Neanderthals and Denisovans overlapped there and in what they were doing there. Yeah, that would be it for me.
That would be amazing. What's your favorite way to relax after a long day?
Oh, I read. I love reading. I love to read. I love to listen to podcasts. I have a large collection of podcasts. And sometimes a little bit of gardening, walking with my dog.
That sounds lovely. I got two more. What is something you've always wanted to try but never ended up doing?
I would love to learn to play the cello. And I've never done that. I've never tried. I think it would be fun.
And one word to describe your life.
Happy.
That's beautiful. I love that. Then my last proper question is going to be one that another guest of this podcast left for you without knowing
nice.
who they left it for. And I'm going to ask you after this recording to do the same.
Okay.
Which advice would you give to early career scholars, for example, master students or PhD students interested in human evolution? Just do it.
Yeah. So I think that the best advice, it wasn't really even advice. So my PhD advisor was very inspirational for me. I did my PhD in South Africa with Professor Winston Hyde. And he just had this tremendous faith that I could do anything, even things that I was quite convinced I couldn't do. And at some point, I just realized I should just take all these opportunities. He offered a lot of opportunities constantly for that took me way outside my comfort zone. And I decided to accept the challenges that he offered, essentially, because he was like, "Oh, you can go and teach this course." And I was like, "But I don't know enough to teach a course." And of course, he would say, "Well, you should just do it." And I'd have to learn enough to teach the course and then do it. And then it was good. So I think that my advice would be take the opportunities you're offered, find a good mentor, find someone who's going to open doors for you and who will support you from behind quietly, as Win did for me, by promoting you and giving you opportunities. And when you get those opportunities, just do it. Don't turn them down. Don't be afraid, and don't doubt that you can. I guess I still always doubt that I can do some things that I'm asked to do, and then I do them anyway. And I've never regretted that.
That's beautiful. I love that. Stepping out of your comfort zone goes hand in hand with a little bit of pain.
A lot.
Yeah, probably. But that's growth.
It opens doors. You meet new people. You learn what you don't like at the minimum, what things you don't enjoy. And at the end of the day, I really think it's true that you mostly don't regret the things you tried, and you always regret the things you didn't do, things you should have tried or could have tried or maybe wanted to try but were too afraid.
Where can people find you online?
So you can find me on almost any social media. Instagram, Twitter, Blue Sky, Mastodon. The webpage of the Institute, of MPI IFA, it's called. You can find my group webpage there.
I'll put all of that into the description. And then or just Google Janet. And I'm sure
you should be able to find most things. I'm not super active on social media, but I do watch and answer questions. So if people have questions, I'm happy to chat.
And to wrap this up, is there anything, Janet, you feel like you want to add, or I didn't ask you? Anything you would like to say?
No, I don't think there's anything else. I think we did a good survey over 20 years of just doing it.
Of just doing it. I love that.
It was fun.
Could go on and on, but
thank
you for your time. Thank you for chatting with us today for the HEAS podcast.
It's a complete pleasure. Thank you very much for the great thought-provoking questions. I'm still worrying about what I'm good at. Now I'm really like, You
did a great job. Thank you so much. Welcome to the HEAS podcast, Jenny.
Hi.
We're thrilled to have you here today, honestly. Could you please introduce yourself and your research to us?
Sure. My name is Jenny Tung. I am the director of the Department of Primate Behavior and Evolution at the Max Planck Institute for Evolutionary Anthropology in Germany, and also a professor of evolutionary anthropology and biology at Duke University in the United States. I study primate genetics and behavior in a variety of different contexts. My group and I are interested in how social and behavioral processes are translated into how the genome functions, and also into the long-term patterns of genetic variation that we observe in the species that we study.
So you're studying living primates and not ancient ones, as you mentioned today during your talk.
That's right.
And more specifically, baboons in Kenya at the moment. Mm-hmm. And actually, you mentioned that you just arrived from Kenya yesterday.
Yeah, that's right. That's right.
That's insane. So welcome to Vienna, first of all.
Thank you.
And this particular study that you talked about has been going on for incredible 53 years. And can you give us an idea how one studies wild primates? And by that, I mean, like, are you just observing them, or are you interacting in any way, shape, or form? And if you're interacting, how cooperative are they?
Yeah, sure. So I am very lucky to be part of this long-term field study that you mentioned. It's called the Amboseli Baboon Research Project. And was founded in 1971 by Jean Altman and Stuart Altman. And Jean was the PhD advisor of my PhD advisor, Susan Alberts, who is also the PhD advisor of the currently third co-director of the project, Beth Archie. So we've been able to do this, I guess, over time as kind of a matriline, which is very similar to the structure of how baboon society works. The way that we study the animals in the wild, so the population we study is a wild population. There is no fence. They're free to come and go and move inside our study populations or leave the study populations as they wish. They are not food supplemented. They are subject to the pressures from natural predators and from variation in the environment from year to year and season to season. So that's what we really mean by sort of a study of a natural population. The way that we do it follows methods that are quite typical for studying living primates, for studying animals in this sort of context of long-term field studies. And basically, that's by a policy of no intervention. So one of the really key pieces of being able to do what we do is that the animals that we study, the baboons that we study in Kenya, don't react to us one way or the other. That is, they're not scared of us, they don't run away from us, they don't see us as a source of threat, nor do they see us as a source of opportunity. We're not feeding them, they're not coming up to us and trying to get food or water or other kinds of resources. So really our goal when we're out there in the field, and this is a goal that's been pretty successful over the decades, is for the baboons to think that we're no different from a tree. And in doing so, we do move, so a moving tree. In doing so, what we're trying to do is observe the ins and outs and the ups and downs of their lives as they live them naturally, as they interact with one another across the days and the weeks and then the seasons and the years, and as we see them have offspring and those offspring grow up and have their own kids and so on. So that's really the key bit is to fade into the background so that we can see what really goes on and what matters in sort of their life outcomes.
So do they not acknowledge your presence at all?
That would be the ideal scenario, is that they don't acknowledge our presence at all. Of course, we have some variation in our population, especially so the particular species that I study, females stay in their social groups throughout their whole lives typically, and males can move around. So if we have individuals come into our group from outside of our study population, they don't know us so well, we learn to give them plenty of space because this process, it's called habituation, is really important to us. And if we see animals who need work on habituation, we're sure to respond to that.
And another question from me would be, which I'm not trained at all, my eye is not trained to tell apart those animals.
Yeah.
Do you learn that, or are they really different from one another?
Oh, right. That's a great question. Sorry, I just overlooked that part. Yeah, so one of the other key things, other than habituation, is that this is an individual-centered field study, meaning that we recognize, and in our case, we recognize on site, and here I'm really talking about the royal we, so I should give major credit here to our senior field team. We have a permanent staff of senior researchers, all of whom are Kenyan, at the site. So we, the royal we, learn each and every one of the individuals in our population on site. And although you say, well, that seems hard to me, I will say that humans are actually very, very good at that. We're very, very good at facial recognition and in learning to tell the differences from one another and to learn the differences between the nature of our relationships with someone who looks like X and someone who looks like Y, right? So what you'll notice if you ever come to our site and you try to learn the IDs of our animals, we typically follow about 250 to 300 animals at any given time. So it's not a small task, but if you learn...
Oh my gosh.
Yeah, but I bet you could do it. The larger primates are not so hard. They are different. They carry themselves differently. Their faces look different. Our observers talk about family resemblances among our animals as well from families they've watched across generations. But if you ask them, can you tell me, explain to me what Abby looks like, or explain to me what rodeo looks like, they would probably stop, they would help you, they would look at you a little funny, and then they'd try really hard to explain to you how they know those animals. But really, it's kind of this phase change where you go from, okay, that individual has darker hair and her tail is kind of kinked and her eyes are close set, to exactly what we do when we interact with other humans. So-and-so looks like so-and-so because that's so-and-so, and I know them. And I think that's where our observers really get to eventually.
That's fascinating. But actually, now that you're saying that, I'm like, yeah, of course there is a big difference to humans, but why would you not recognize somebody's face? And if you're working with them so closely together over such a long period of time, of course you would start recognizing facial patterns and how they carry themselves, as you said.
Yeah.
That's pretty cool. So your lab is particularly interested in the interplay between genes and behavior of primates. Mm-hmm. In baboons, social relationships are amongst the most important predictors of survival and not only the genetic makeup, as some people might think. Mm-hmm. Can you explain how social bonds are influencing survival, and does that in some way also apply to humans?
Yeah, so you just asked the $1,000,000 question. So some of my colleagues who work in the social sciences, when we talk about our shared interest in social relationships, health, and survival, it's often called the social determinants of health, or part of the social determinants of health in humans. They will point out to me that the findings that our project and other studies of wild social mammals have produced in the last 10 or 15 years or so are, as a pattern, something that they've known about in humans for over a century, right? That social relationships, social bonds, social support predict how healthy people are and how long they live. And so we're really playing catch up in these other species and hopefully providing some insight that this sort of relationship between social ties on one hand and health and mortality outcomes on the other in humans probably has much deeper evolutionary roots in our lineage. So we are providing that, but I'll just point out that pattern, as they remind me, sociologists have known that for over 100 years. And so when you say, how does it do it? Well, that's exactly the question. And so for us, the relationship between social affiliation, social bonds, and survival is something that we've actually only known for more like a decade or so, because it takes a very long time in even the longest running long-term field studies to watch individuals grow up, experience different environments, and die. So it takes decades worth of data to just be able to say that. We've been very interested subsequently in what on earth could be doing. And I suspect that what we're seeing is the confluence of many different mechanisms, many different pathways through which social bonds, social affiliation can influence survival, right? You can think about it in some simple regards. In meerkats, for example, which is another species that we do some work on in my department, this is not a primate, but it's another highly cooperative social mammal. Individuals who are more peripheral from the group die at higher rates, probably because they're getting preyed on. So that's one reason, right? Having close social bonds, a lot of social tolerance, being close to each other, that probably has an effect on whether predators can get you. In Barbary macaques, another social primate, individuals who have stronger social ties benefit from thermoregulatory benefits. That is, they stay warm together, they huddle together. And so there's a direct reason why social ties might matter. In other cases, social bonds probably support coalitionary behavior between individuals within the same group. That is, you have an ally in fights with other individuals. And that probably matters. And so I think as more and more work is being done on trying to explain what links these sort of social patterns on one hand to these survival outcomes on the other, what we're finding is that there are probably a lot of different pathways, and they matter differently depending on the causes of death that are important in that population. And that's probably, although less satisfying than a single mechanism, what's going on in humans as well.
I was going to ask you about humans. A lot of these pressures that you just mentioned, like predators and stuff in the wild, are actually removed from us humans in our contexts and worlds.
Yeah.
So how does that still apply?
Yeah. So I think that predator-related explanations are really only one possible mechanism through which these kinds of links can be established. We know, for instance, that differences in the strength and quality and quantity of social relationships in the baboons that we study also have more direct molecular and physiological effects. So, for example, animals who have stronger social bonds tend to have lower levels of glucocorticoids. This is a hormone that is associated with the stress response and which, if elevated over time, can have both metabolic and immune consequences for the body. For example, they can dampen immune defenses, or they can actually lead, perhaps counterintuitively, to uncontrolled inflammation over time. And so it's not just that there are external forces that interact, external threats like predators that interact with social relationships and risk. There are also internal changes to what's going on, some people say it, under the skin in the animals that we're watching.
A very interesting aspect of your talk today was the presence of a mother, especially during the early life stages. That seems to be very crucial and impactful on overall health and survival and mortality in these individuals. And it also plays a role in how well they are then doing in the raising of their own offspring.
Yeah.
Could you tell us more about that, please?
Sure. So baboons, rather like humans, have very dependent, very helpless babies. Sometimes people who study animals will talk about the difference between precocial animals and altricial animals. So these are terms that basically reflect the distinction between species, between babies. You know, if you've seen nature documentaries, sometimes you see baby antelope being born and then they stand up and they start walking right away. And for most of us who've had any experience with human babies, that does not happen for some time, right? Babies are very, very dependent on help from adults in our species and in many other primates for quite some time before they're able to forage and move around on their own. And so most of that support in baboons comes from mothers. We've seen a few cases of putative adoption by other females when a mother is gone, but these are very, very rare, and honestly, they're often not successful in terms of how the kid does. So mom is this incredibly important figure in the lives of baby baboons. And you can think of that for one very direct reason when they're truly infants, which is that mom is the only source of nutrition before babies are weaned. And so that is quite important, but maybe kind of trivial in terms of what we would expect. Basically, in our population, if mothers die before those infants reach about a year old, when they're starting to do some foraging on their own, we almost never see the babies survive. What is perhaps more interesting, though, is that kids are dependent upon their moms for much longer than that. If they lose their mothers when they're 2, when they're 3, when they're completely nutritionally independent from mother's milk, they still are very responsive to the presence of their mother in terms of whether they make it. And for those kids who lose their mothers, anytime in that sort of infant and juvenile period, we find that their adult lifespans, when they're well independent of their mothers, when they might be having kids of their own, when they're moving up in the world as full adults, are still truncated if they lost their moms early in life. So, we think that this is coming from a variety of things. Mom may not be feeding their kids directly, but she certainly is helping to give them examples of what foods to eat and how to process them. And she's certainly doing a lot to help introduce them into the social environments that they have to navigate later in life, including for daughters, helping those daughters figure out where they fall in the social hierarchy of the group.
As you were explaining this to me, I always thought, are we still only talking about baboons? Because that applies to us humans so well.
Yeah.
Also. And there is the grandmother hypothesis in humans. That's please correct me if I'm wrong. It's been a while since I heard that in my master's. And it's a grandmother that takes care of the offspring as well so then the offspring is just doing better. Do we see something like that in baboons as well, or in other primates, that the grandmother might chime in and support?
Yeah, that's a really interesting hypothesis that was originally proposed to help explain this very puzzling thing that humans do. So now we are getting to something that's a little bit, but not exclusively human specific, which is to stop reproducing when there's still a very, very long time that they can still live. We're talking about menopause, particularly in human women. And as far as we can tell, in baboons, there is no sort of routine menopause. There are some females who sort of, lose reproductive condition towards the end of their lives, especially if they live very long lives. But females tend to still be producing their own offspring even later into life. So the sort of conditions where those females might be helping with care of their daughters' kids aren't really in place. However, I'm not the right person to ask about this because it wasn't my work, but there was a really fascinating paper that came out earlier this year or at the end of last year on the potential for menopause or menopause-like early reproductive cessation in one chimpanzee population that's been followed in Uganda for quite some time. So it may be possible there, and I think obviously an interesting question would be the degree to which females who stop reproducing are beneficial to their grand offspring survival. I think that would be very fascinating. And in fact, it has also been something that's been described in orcas and some other cetaceans for some time.
Thanks for educating me on that. You explained it so much better than I could. Okay, now let's move on to our next segment, which are the listener questions from our audience.
Great.
The first question is, based on your experience, what plays the bigger role in the life and health of primates, the genetic makeup or the social structure?
Oh, okay. So I'm going to come down hard on it's the social environment here.
Yeah.
Yeah. So what we've seen are these just very, very large effects of social bonds and early life adversity, and especially for fertility-related outcomes and mate competition, aspects of social status on how well animals do. And while an individual's interest in forming social relationships or their capacity to form social relationships may be in part heritable, may in part have a partial genetic component. We have one result that suggests that how much they groom is partially heritable. Most of that, I think, is determined by the groups that they are born in. Do they have a lot of opportunity for social partners, right? Do they have a lot of age mates? Do they have a lot of family in that group? Is their mother good at that kind of thing? Do they have examples where there are many grooming relationships that that they are, or sort of relationships between others that they kind of can slot themselves into? So, yeah, I think that if we're talking about the determinants of individual success across the life course, then those kinds of social factors probably are doing much more to explain variation in who does well and who does less well. I say this as a geneticist who's very interested in long-term evolution of social relationships and social behavior as well. Mm-hmm. And I think these things do change and are responsive to natural selection over long periods of time. But on a short-term timescale, if you wanted to point to an individual who is going to do well and an individual who's going to do less well, I think you would much rather know about their sort of social milieu than about their genotype.
Mm. And that question is perfect for you because I'm talking to a geneticist. So I'm not from the field, but genuinely interested. I've been wondering, what's the difference between genetic and genomic?
Yeah, so I think those are squishy terms. I mean, the term, when we talk about genetics, we often are talking about differences in DNA sequence in actual genotype, right? In the actual base pair composition of the genome. We may be talking about how those sequence differences are passed on from one generation to another according to sort of the rules of Mendelian inheritance. We may be talking about how differences in that sequence translate into differences in traits. Sometimes when we talk about genomics, we're talking about something similar, but often people will use genomic in a broader sense to also mean, how does the activity of our genes modulated by the environment, right? That can be a genomic question, or maybe even to refer to the scale of analysis, right? Rather than looking at single places in the genome, you're looking at the whole thing altogether. But to be honest, they are used a little bit squishily, so it's a good question for sure, and a good question to ask anybody who's using those terms if you're unsure.
Thanks for explaining that. Now, our next segment is called the He Has Quick Takes. So I explained to you that's just a rapid fire question round, and feel free to say next or whatever if you don't want to answer one of these. They're not crazy personal. Don't.
I think the last time I had to do something like this was on my qualifying exams as a PhD student. I had a lightning round. Yes. So it can't be as bad as that. So please go ahead.
Exactly. No, I'm not trying to trigger you.
No worries.
They're the easygoing questions. For example, on your days off, are you an early bird or do you prefer sleeping in?
Depends if I'm in the field or not. If I'm in the field, it's really, really hard to sleep past 6:30 or so. There's a lot of birds going on and you're living outside. If I'm at home, my son, who likes to sneak up before me and watch TV, would probably tell you that I like to sleep in a little bit.
What's your favorite food?
I love food. So that one's actually quite hard. But I would say there's quite a lot of different types of seafood that fall into my favorite food group.
I love that too. 3 words to describe your job.
3 words to describe my job. This one's really hard. Let's see. Why is that so hard? It's varied. So I never do the same thing on any given day. I will say fortunate because I feel incredibly lucky to do what I do and get to interact with the people that I work with and to be able to work in the settings I'm allowed to travel to. Unpredictable. I suppose that's related to variable, but I also mean that from a scientific sense. We often start projects and we think we know what we're going to see, and our results and the real world take us places that we didn't expect, and that's part of the fun.
Sounds like an amazing job. What do you do to motivate yourself?
What do I do to motivate myself? Anytime I get the chance to interact with interesting, smart people, whether they're students of mine or collaborators that I see on visits like this, I find them actually sometimes immediately very tiring in the sense that I need to take a nap after all the interaction, but intellectually extremely exciting and motivating. They always give me sort of ideas and different perspectives on things that we can do. And so it's funny because probably on balance, I'm a bit of an introvert, but I get a lot of energy and motivation from talking to other scientists.
It's so interesting that you're mentioning that right now because my last guest was Janet Kelso, and we talked exactly about that. I asked her in the rapid fire questions, are you an introvert or an extrovert?
Yeah.
And she also said, yeah, of course, don't get me wrong. I love speaking to people, but then I need a nap.
Yeah.
Like, yeah, right. I feel that.
Yeah, Janet is a colleague and a friend, and maybe that's not accidental.
Oh, she's so lovely. It was such a pleasure to be chatting with her. We really had such a good time. And I'm also having such a great time with you. I could speak to you for hours. I really had to intentionally cut the interview segment short because, I would go crazy on there.
Thank you.
2 more questions,
Okay.
and then we're done with that segment.
Yep.
If you could have any superpower, what would it be?
Oh, wow. Any superpower. I feel like any superpower you wish for, of course, there's some secret catch to it that will make you regret it. But, being able to go back and forth in time, I think would be incredibly cool, both for like small scale practical reasons, like getting more sleep, and also because, there's so many things that we can't see that we're just trying to infer about what happened in the past or what's going to happen in the future. And wouldn't it be cool to take our best guesses about how our study species evolved and then go see it? My study population in Amboseli is in an old Pleistocene lake bed, and I would love to see what that place looked like, thousands of years ago. So yeah, probably that one. Even though I'm sure there's a catch.
And last one, what's your favorite mantra or quote?
Oh.
I love how I promised you easygoing questions, and then I come in with those.
I'm not sure. I'm not sure I've got one. I honestly feel like I probably repeat myself a lot when I'm talking to my students, and so they might be able to come up with something that Jenny says all the time. But I don't think I have a mantra. So I might have to give that one a pass.
All right. Then let's move on to the question that one of our speakers, one of our guests on this podcast left for you. And I like that question. Which part of your job do you enjoy the most?
Oh, the discovery part. I actually like a lot of parts of my job, and I like a lot of parts of my job, but I wouldn't do my job because of those parts of the job. Mm-hmm. The reason that all of the sort of support work that we do, all of the sort of bureaucracy and logistics and management and permitting is ultimately rewarding is because it's in the service of learning something new about how the world works that we didn't know before. And so that's the best part. I've been spending much of the last year and a half or so trying to set up a new group and a new department in Leipzig, which, again, to go back to something we talked about earlier, I feel incredibly fortunate to be able to do. And I can see just now the first sort of glimmers of new discovery, and that reminds me why we do it all. So yeah, that one's an easy one.
That, for example, motivates me to hear that from you. And also congrats on that, because you're not only the director of that department, you're also the founder of it, right?
Yeah, every director has found something new. So yeah.
Yeah, it's incredible.
Thanks.
No, I looked you up and I'm sincerely impressed by all the research, and now also by your personality, because now I got to talk to you, and by your talk and everything. So that's definitely, you are motivating people out there, and I'm really happy I got to sit down with you today. And if people want to know more about you, where can they find you online?
You can look for information about me and my department at the website of the Max Planck Institute for Evolutionary Anthropology. You can find out more about the Baboon Project at the website for the Amboseli Baboon Research Project. I'm on Twitter, but not terribly active, at jtung5.
And Jenny, is there anything you would like to add to today's conversation, or anything you feel like I didn't ask you?
I think you did a fantastic job, and I'm very thankful for the opportunity to sit down with you today.
Thank you so much for sitting down with me for the HEAS podcast today. Hello again, Victoria here. If you enjoyed this episode or the podcast in general, please consider showing us your support by liking, sharing, and subscribing to our podcast on Spotify and YouTube. Your feedback and engagement mean the world to us and keep us going. Thank you for joining us on this journey, and stay tuned for the next episode of the HEAS podcast.